Hello guys, selamat sejahtera. The title of my journal is Evaluation of Probiotic Potential of Lactic Acid Bacteria Isolated from Traditional Malaysian Fermented Bambangan, which is Mangifera Pajang, and the author of this journal is Seah and then Siew and Birdie Scott and Fook. So guys, basically this document discusses a study that evaluated the probiotic potential of lactic acid bacteria, which is isolated from this traditional Malaysian fermented Bambangan, which is Mangifera Pajang, and the LAB were tested for their tolerance to pH, bile salt, and antimicrobial potential, and their auto-aggregation ability, and then microbial adhesion to solvents, tolerance to high temperature, and osmotic pressure. So the study found out that approximately around 36% of these isolated LAB strains were displayed excellent survival at pH 3 with at least 4 log colony forming unit per milliliter after 24 hours at 2% of bile salt. Additionally, the LAB strains showed high aggregation activity with around 5 lactobacillus plantarum strains exhibiting at least 70% viability at 60 degrees Celsius for 10 minutes. And then furthermore guys, this study the maximum beta galactosidase activity was found in 4 lactobacillus plantarum strains, and this study concluded that these LAB strains could serve as a promising probiotic candidates for the preparation of functional food products. And the study also identified around 28 LAB strains, primarily Lactobacillus plantarum, Lactobacillus brevis, and Lactobacillus rhamnus, with high survival rates at low pH and bile salt concentration. These strains were evaluated for their carbon utilization patterns, and they were found to possess a different metabolic profiles. The LAB strains were also tested for their ability to survive in our GIT, and the most strains showing stable viability in the intestinal juice at pH 8. Moreover, the LAB strains demonstrated high auto aggregation ability, an important property for adhesion to the intestinal cell wall. Additionally, some strains exhibited high hydrophobicity, indicating a strong capacity to adhere to intestinal cells and solid materials. And then furthermore, the study investigated the inhibitory activities of the LAB strains against common foodborne pathogens, and the results showed a varying degrees of inhibition against the Staphylococcus aureus, Listeria monocytogenes, and Salmonella typhimurium, and Salmonella enteritidis, and Yersinia enterocolitica. And the findings suggest that these LAB strains possesses potential antimicrobial properties. And then as a conclusion, the study provides a valuable insights into probiotic potential and technological properties of isolated LAB from traditional Malaysian fermented Bambangan, and the identified LAB strains, particularly like Lactobacillus plantarum, demonstrate promising probiotic characteristic, and this could be utilized in development of future food product. And these are the overall research contributes and my understanding on the journal that I've chosen. My personal feedback or critics on these journals are, the strength of the journal is that evaluation of probiotic potential of LAB isolated from traditional Malaysian fermented Bambangan, include the comprehensive identification and characterization of lactic acid bacteria strains, particularly Lactobacillus plantarum, Lactobacillus brevis, and Lactobacillus rhamnosus, with their high survival rate at low pH and bile salt concentration. And the study also provides us a valuable insight into probiotic potential and then technology properties of the isolated LAB strains, and demonstrating their potential application, as I mentioned earlier, in their development of functional food products. And then additionally, this research was supported by the Ministry of Education Malaysia, indicating a strong foundation for the study. As a nutshell, this study could be expanded to explore more potential health benefits and functional properties of the identified LAB strains in animal models and human subjects. And then future research could focus on the development of novel probiotic products using the stress tolerant LAB strains. This is the overall understanding of me from the journal that I've chosen. And thank you for having me here, and thank you for your time. Thank you so much.